Hello and welcome. I am Shanice Battle, your moderator for today's discussion. I recently graduated from the School of Public Health with a PhD in epidemiology this spring, where my research focused on measuring exposure to neighborhood structural factors and exploring their association with mental health outcomes among U.S. adults. I'm here today with three faculty experts from the School of Public Health, representing three of our six departments, and I'll let them introduce themselves now.
Hi, everyone. My name is Belinda Needham. I'm an associate professor in the Department of Epidemiology, and I co-chair the Center for Social Epidemiology and Population Health. My research focuses on the biological mechanisms that link social factors to health.
Hi, everyone, and thanks, Shanice. My name is Dana Dolinoy, and I'm a toxicologist that studies how exposures affect our health. And I also serve as the NSF International Chair of Environmental Health Sciences and direct the Michigan Life Stage Environmental Exposure and Disease Center. Welcome.
Hi, everyone. My name is Edward Ruiz-Narvaez. I am associate professor at the Department of Nutritional Sciences at the School of Public Health. My research is focused on trying to understand the genetic basis of complex diseases in human populations, in particular in minority populations. Thank you and welcome.
Okay, thank you all for being here today. We're going to go ahead and get started. So today's discussion is about why science changes and why that is a good thing. The Spring 2021 findings issue focuses on the idea that throughout history, the process of discovery has always involved correcting mistakes, clarifying our understanding, and adding a deeper shade of nuance. For example, astronomers thought they had pinned down the position of the Earth relative to the sun and planets until new information emerged that led to rearranging the model. These changes in our understanding are features of science, not bugs. The world changes, society changes, and so science changes. Science is one way we interpret the world, so it helps us navigate those shifts, and by embracing new scientific insights, we can lead healthier, more informed lives. With flexibility and fluidity being so central to science, we need a shared understanding of the process of how science changes and why it benefits us all. So to kick off our discussion today, I want to talk a little bit about why we see scientific change. What changes in science related to the pandemic might demonstrate this? And Belinda, given your unique positioning as a professor in the epidemiology department, I'd love to hear some of your thoughts.
Sure, thanks, Shanice. I'd be happy to talk about that. So, over time, our understanding of a scientific question or problem might change because our understanding improves. Maybe we have more data about something, or we have better data to understand a question. And I think COVID-19 provides a really great example of that. So, a year and a half ago, none of us had heard of COVID-19. It was a brand new thing. And so it took a while for us to gather the data that we needed to understand things like how the disease is transmitted. So you might remember there was sort of a big debate for a long time about whether it's transmitted primarily through droplets versus aerosols. So is this an airborne virus? And we had to collect data to understand, which was more important, right? And as we learned more about the transmission of the virus, we saw that the public health recommendations changed and sort of, we shifted from sort of emphasizing things like disinfecting surfaces toward really more emphasizing the wearing of masks. So that's one thing that happens is we get more data, we know more about a problem. Another thing that contributes to changes in scientific claims is that, we might have a new scientist or a new group of scientists who come to a research problem with a totally new way of looking at the evidence that's already there. And so you might see that there's sort of a paradigm shift and not, so our evidence might not change, but our theories might change. And so there's sort of many ways that scientific claims might change over time.
Yes, I love how you commented on changes as improvements in our understanding and how as we gather data, we can then adjust our public health recommendations. I mean, imagine if we stopped at, hey, you don't need to wear a mask, just disinfect surfaces and you'll be fine. Imagine where we would be. And also thinking about this idea of new perspectives, and as new scientists enter a field, they bring new perspectives and new theories. Speaking of new perspectives, Edward or Dana, would you all like to chime in on this?
Yeah,
Yes.
thanks, Shani. Oh, go ahead, Edward.
Sorry for interrupting. Yes, I want to add to what Belinda said before, and I think we need to see the scientific change and as a strength of the scientific enterprise. And this question that we have right now during COVID pandemic, and I think that reflects that we're trying to understand and to solve a problem. And then we need to see the changes as a strength, and also this new perspective that you mentioned before, because not only science changes, we need to remember that science is part of the society, and also society changes. And the expectations of society about science also is going to change over time. Then, I'm thinking about the pandemics. Now we see, or at least we are learning that these pandemics have affected health in multiple levels. For example, mental health or the economic crisis. There is pandemic fatigue in the population. And now the society expects that scientists are going to at least to advise and to provide some guidelines how to navigate this new scenario that we have. That imply that scientists are going to work, for example, with policies making, with economists, with different branches of science. Then we need to see this change and the strength of the scientific enterprise, definitely.
Yeah, thanks, Edward. I agree. I think that with emerging scientific issues like COVID and so many things that we study as scientists, it can be really hard to grasp that not only are there things that we don't know, but there are things that we think we do know that end up being wrong. The generation of scientific knowledge is not a straight line. And as we get new data, as Belinda indicated, or sometimes totally new technologies, good science can evolve into something else. And as we've discussed, we saw this early on in the pandemic when we thought surface contamination was a main route of exposure. And all of us were wiping our groceries with sometimes very toxic wipes that contain, ironically, chemicals that can affect our health through mimicking or blocking endocrine system. The pandemic also forced us to take a harder look at environmental racism. These are the systems that produce and perpetuate inequalities in exposures to environmental chemicals, things like air pollution or toxicants in our diet. These systems include indifference or ignorance, inadequate testing of our industrial chemicals, or housing discrimination, for example. But through this, by anticipating that some groups are facing outsized environmental exposures, we can now act to protect lives, not only during the end of this pandemic, but thinking about future outbreaks.
Those such rich answers there. I don't know what to touch on first. First, Edward, you brought up these long-lasting impacts of COVID and how we still have a lot more to learn and a lot more is going to change before it stops changing, if that ever happens. Especially thinking about the way that COVID is going to have an impact on various aspects of society, and society is changing as a result of the pandemic, in addition to the changes that normally happen in any society. And Dana, I love how you mentioned that emerging focus on structural racism, right? And environmental racism. I remember reading an article sometime last year by John Zellner at the School of Public Health, and he described pandemics as a litmus test for inequality. So it really lets you know where the equality lies and how exactly, really shows you how that can impact health. So great answers to those questions. Our second question today is, what's an example from your own research of how your thinking on a particular issue or topic changed, and you now view or study something differently because of it? Dana, I'm thinking of some of that groundbreaking research you did as a PhD student, which led to the creation of the field of environmental epigenetics. So I would love to hear from you on this question.
Oh, thanks, Shanice. Yeah, I mean, I think it stems all the way back to the late 1990s, early 2000s, when we saw the great public-private partnership that was going to sequence and did sequence the human genome. So at the beginning of the Human Genome Project, we thought we would find about 100,000 human genes. But by the end, it turned out that we only have about 20,000 human genes. But there's still a lot of hope. We thought when we sequence the genome, we'll identify the genetic sequences that help determine how people remain healthy or why they get sick. But it turns out that there's a whole second genome called the epigenome. An epigenome literally means in addition to or above the genome. In fact, it's the instruction book that tells these some 20,000 genes when to turn on, where to turn on, and how to react to various environmental influences. And by environmental influences, we're very inclusive. I study toxicants and how nutrition interacts with those toxicants, but you'll hear a lot today from my colleagues who study things like social stress. And all of these things can get under our skin and affect our gene through the epigenome. And so it was back in the early 2000s that we were using a really cool mouse model called the viable yellow agouti mice. And they range in coat color from completely normal brown to mottled to completely yellow. And the yellow mice become obese as they get older. And what we saw is that the mother's diet could influence the percentages of brown lean mice or yellow obese mice. So what was really interesting is when we first looked at a toxicant, and we looked at a toxicant that many people are aware of called bisphenol A, or BPA for short. It's a monomer that makes up all the hard, clear plastic. And in fact, it can interfere with our endocrine disrupting system. But when the mouse mothers were fed diets high in bisphenol A, we saw that bisphenol A didn't change the offspring genome at all. There were no genetic mutations, but there were lots more yellow obese mice. And this was due to an epigenetic change in the agouti gene that caused that gene to be on all the time. And those mice don't feel full and they continue to eat. So that was a really interesting finding. But unlike the genome, which is static and not modifiable, we can't do a lot to change our genes. The epigenome is dynamic and plastic. And so we performed a second experiment. And this time, the mothers were still exposed to BPA, but they were given a diet high in methyl donors like folic acid, methionine. These are things found in green leafy vegetables, or the phytoestrogen called genistein, which is found in soy. And in each of those experiments, if the mom had a good diet on board, that reversed the effect of the bisphenol A alone. So this gave us some hope that we don't have to settle for the genetic hand that we're dealt, but that we can use environmental influences, including nutrition, to counteract the negative effects of toxicants that we encounter throughout our lives.
Yeah, so I'm imagining being a PhD student, and coming across something that's conflicting with everyone else in your field, or some of the longstanding ideas in your field. So what were you mindful of as you reported this new finding or perspective?
What's really interesting about that story is this was supposed to just be a 3-month rotation project. And then I was supposed to go back to a different lab and study environmental justice and lead exposure. So once I discovered this field, which was just now beginning to be started, this toxicoepigenetics, environmental epigenetics field, it became a really important way to translate our science to the population. So, this was done in a rodent model. Humans are not rodents, but we have genes that act like the agouti gene. And so anytime we have a variability in an outcome, such as asthma, you can have severe or more mild asthma, it may be due to the fact that we just have underlying differences in our epigenome. And the environment can come along and push it one way or the other, so that you can push someone who might have had mild asthma into a much more severe asthma just by having an environmental trigger on board during development.
Wow, such cool research. And Edward, I know you talk a lot about humility in science in your research. Is there an example from your research on how your thinking on a particular issue or topic changed?
Yes. Also, I want then to add to what Dana said before.
Of course.
Related also to this humility that we need to have, because at the same time that Dana mentioned the late 90s, early 2000s, researchers were starting to sequence the whole genome. And I remember if you read scientific journal at that time, what is the editorial saying that we're now going to know what is it signifies to be humans, what is meant to be humans, thinking that the information that DNA sequence in some house is going to answer the question what is to be a human. But and then part of my research is to try to understand the genetic basis, what are the genetic factors related to disease, to different health outcomes. But what we have been learning is that we still don't know, in most of the cases, how variation on the DNA sequence affect health outcome. I think what is this last year of research have telling us is that we need to be humble. And now how Dana mentioned before, the environment has a huge importance in our health, different health outcomes. We need to consider, and by environment, I mean diet, pollution, even our fetal environment when we are developing in uterus. But the problem that I see that even that right now we know the genetic factors by themselves, they are not going to determine our health outcome. Still, I think there are theoretical limitations in our approaches, because still there is a genetic determinism that is implicit in our models. We need to take into account the environment. Of course, some of the limitations that we have to study environment are practicals. Let's say, for example, to evaluate interaction between genes and diet or any type of environment, we need large sample sizes, and we have those limitations. But some of the other limitations are also theoretical, because we need new model, we need new thinking, and to be humble, to be aware that, at least in my field, that genetic determinism is rather a limitation of our understanding of reality. And also, I want to add the related to genetics, that most of the studies have been carried out or have been done in white population. There is an underrepresentation of minority population, Hispanics, Blacks, African Americans. Then also still we don't know a lot about the genetics of diseases in minority population, and this is an important issue that we have to address.
Shanice, I think you're muted.
Oh, there I am. Thank you.
No problem.
Again, really great answers here. And I really like that you mentioned this perspective of sometimes we just don't know. And that's kind of that humility that you always have to approach science with. And even if we arrive at an understanding of something, it is subject to change at any point in time in the future. And Belinda, did you want to add anything to this question?
Yeah, I'll just say a couple of things. One thing about sort of epigenetics and that field, and then I'll also talk about another example in my work in telomere that I think relates to this question. But I was just, as Dana was saying, rats aren't people. that is true. And now there's this field that's sort of emerged in the past five or so years of social epigenomics, where people are trying to translate some of these findings that we've seen in animal models and looking at humans. And so, I've been doing some of that work. So I've done some work in the multi-ethnic study of atherosclerosis, which gets at Edward's point about a lot of this work not being done in multi-ethnic populations. And so we looked at how life course socioeconomic status relates to methylation in genes related to stress reactivity and inflammation. And then I'm also doing some more work now in the NHANES study, the National Health and Nutrition Examination Study, where we're getting genome-wide DNA methylation for about 2,500 people in a nationally representative sample to help understand the extent to which sort of DNA methylation patterns can help us understand racial disparities in cardiovascular morbidity and mortality. So I think that's a nice example of how these sort of different areas of research build on one another and are helping contribute to our understanding. But I also wanted to say a little something about the telomere research, because that was talked about in the article. So telomere length is a biomarker of stress and cell aging. And when I came to this field a little over a decade ago, the sort of conventional wisdom was that longer telomeres are better. And you might have even seen commercials selling telomerase supplements to lengthen telomeres. But now we know from additional research, including Mendelian randomization studies, that long telomere length is a risk factor for several kinds of cancer. So we weren't entirely correct thinking that longer telomeres are always better. And so, the humility that Edward talked about is very important there. I don't think that these supplements really work, but if they did, it could potentially be dangerous, right? And so, we need to really think carefully about whether we have enough evidence to be making recommendations for things like supplements or treatments.
Definitely. Awesome. Great work you all are doing.
Shanice, may I ask Belinda
Sure.
a question about the telomeres? I think that your work is fascinating. Is it right to sort of think of this as telomeres as sort of like a Goldilocks thing where you have to have just right? Like, so too much is bad, too long is bad, and that leads to cancer, and too little is bad, and that can lead to other types of negative health impacts, because we see that a lot in the fields of nutrition and toxicology, where everybody assumes like different linear lines. So in toxicology, we assume like more toxicants are bad, but there's a phenomena where these low dose chemicals can actually be worse than high doses of chemicals. And then Edward, the same thing happens in nutritional sciences, right? Where we assume that things like folic acid, which is a methyl donor, that's a good thing. But in fact, if you get too much folic acid, we might methylate our epigenome in a way that is not positive. So we're always like trying to find that Goldilocks just right.
Exactly, and I think that's part of our also going back to the humility that we talk about. There are things that we don't know. That we don't know what is the right spot, let's say, for some particular diet or exposure to certain environmental factors, and that's part of the scientific enterprise.
Yeah, and Dana, I just wanted to respond to what you were saying. I think it is true that there's probably a healthy range for telomeres, and that there are these sort of pathological extremes, but for most of us, there's going to be this healthy range. But one of the things that's been tricky for establishing that in the population is challenges with measurement of telomere length outside of the clinical setting. And so right now we have a lot of pretty noisy measurements that make it hard to say this is if I measured my telomeres, it wouldn't really mean anything, right? Unless they were extremely short or extremely long. So we don't totally know yet for the population what counts as healthy.
Great. Okay. And Dana, we have a question for you coming from someone in our audience, building on some of your research regarding those environmental factors and their influence on the epigenome. And the question is, how about socioeconomic mobility? If someone grew up for a while in the inner city, but was able to move to the suburbs, how does this affect the epigenome?
Yeah, that's a great question, because our environment is not static, it changes. It reminds me, we asked a question like this in a study on high altitude from people who live in Cerro de Pasco, Peru. So people who were born at high altitude, we were able to see epigenetic changes. But what happened if they moved down to Lima, which is at sea level? Did those epigenetic changes? And the answer is that some stay and some reverse. And I think that in relation to your question about socioeconomic mobility, the same thing might be true. Epigenetics is a relatively new science, and what we haven't done so far is map the underlying baseline epigenome. So our genome is static. So I can take your DNA, I can take your blood sample and get your genome, and that same blood DNA will be represented in your brain. But our epigenome is different by every cell type in our body, by every organ in our body. And our epigenome also changes over age. We don't want the same sets of genes expressed as when we're 4 years old as when we're 40 years old. But what the data are showing us is that there are some sets of genes that are environmentally labile very early in development, and then they are set. And so I don't think that those would reverse within one generation of socioeconomic mobility. In fact, the field of epigenetics is somewhat controversial because there are some data, and they're mostly in animal models, that show a great-grandmother's exposure can be transmitted 4 generations down into the great-grandchildren. And that is completely mind-blowing, because something that we do now can affect our offspring decades later. Now, that's probably the exception to the rule. And most of what people focus on is the one generational exposure. But whether we can change our epigenome within our single lifetime is something that's still being studied.
Interesting. I mean, this is such cool research. And then thinking about, I mean, I'm hearing all of these, at least in my mind as a researcher, I'm thinking of what you're saying, Dana, and I'm thinking of all of these unanswered questions that still remain in your field. And this brings me to Dean Bowman's article and how he mentions science as being patience and uncertainty, and also science being speaking and listening, and how sometimes the fluidity of science and some of these changes in what we see in science provides an access point for criticism, which leads us to then, or challenges us to balance the social and political influence on science and defense of best practices. So as we think back over the past year and a half, we have definitely seen this balance happening in real time. So I want to hear from you all on how you view the fact that science changes over time. What opportunities would you celebrate, and what pitfalls do you try to avoid? Edward, I'm thinking of your research on genetic factors playing a role in that higher burden of disease and minority populations, and how that has shifted a lot over the course of your time studying. So if anything, would you like to offer any perspective on that?
Yes, thank you for the question. I think that's an important point to consider. The article of Dean Bowman show us society, there is an expectation from society that the answer that science is going to offer, and I think we saw this during the pandemics. But also regarding my research on minority populations, is because now we are, I think COVID has shown us the underlying inequality that in our society, and we need new thinking, new paradigm to try to ameliorate or to solve those problems. Now, my research on minority populations, I'm not only doing genetics, I'm also studying social factors and trying also to do some social epigenomics work, because I think if we really want to improve health at the population level, we need to take this multidisciplinary approach. We need to, as a scientist, to collaborate with different disciplines of scientists, because again, there are questions that still, even we don't know what those questions. We have some questions right now. We may have some answers, but still we don't know what are the questions that we'll be asking in the near future.
Yeah, and then I'm also thinking about minority populations and this idea that science changes, and when you're communicating a change in science, and how some of that mistrust that lies in certain groups is something that is very important to navigate and be mindful of in your messaging. And I know, Belinda, you also have a focus. I know that in the article there was a mention of like this idea of shorter telomeres or longer telomeres meaning a longer life, but then there was a difference in that result when it comes to African American populations. So how do you kind of navigate some of those tensions between the social and the political and science and some of your research? And that question is for anyone.
Yeah, no, that's a really great question. So, when I first started doing this work, I really expected that telomere length was going to sort of help us understand both socioeconomic disparities in age-related disease, but also racial and ethnic disparities. And it turned out that the findings sort of fit our hypothesis pretty neatly for socioeconomic status. So people who had lower levels of education had shorter telomeres, and that helped explain why they had increased risk of mortality and morbidity related to cardiovascular disease. But when we looked at race differences in telomere length, we found that on average, Black people in the US have longer telomeres than white people, which was really very counterintuitive and not what we expected to find. And so, initially I reported it in a paper, but I didn't make a big deal out of it. I thought, well, maybe this is a fluke and it won't be true when, in other data sets or something like that. But it turned out that over time, more people were reporting the same finding. And what I was seeing in the literature is that sort of repeatedly when people would report this, they would say, well, the most likely explanation is a genetic difference. but there was no evidence for that. It just sort of fit within this sort of like essentialist notion of race as a biological factor. And so I found that really frustrating. And the more that I sort of looked into this race difference, I thought, I need to explore this. I need to understand it. Yeah, it doesn't fit with my pre-existing idea of how telomeres fit into health disparities research, but I need to understand it. And so, now I'm doing work bringing in that information about longer telomeres being associated with cancer, and this other sort of thread of research that has looked at how carcinogens are related to telomere length and found that PCBs in particular are associated with longer telomere length. And so now we're trying to bring all those lines of evidence together to understand the extent to which differences in exposure to carcinogens explain the black-white difference in telomere length. And what's interesting, we've talked about environmental racism a little bit already in this, but the reason there's those differences in exposure to PCBs is because of residential segregation and occupational segregation. And so this brings a very different perspective to thinking about that race difference in telomere length, which was an unexpected finding, and moves away from thinking about sort of race as biology and thinking about race as more of a social construction and representing different environmental circumstances.
Yeah, can I add something to that? Yes, I wanted to add also this about these unexpected results that we sometimes get. Working on my genetic research a couple of years ago, maybe 4 to 5 years ago, we did this genome-wide association study. We explored the whole genome to try to see genetic factors related to breast cancer in African American women. The hypothesis was, in particular, this estrogen receptor negative breast cancer is more, the incidence is higher in African American women. Then the hypothesis was that maybe there were certain variants, certain alleles that were more frequent in African ancestry populations compared to, let's say, European ancestry populations. In fact, we found a particular variant associated with higher risk of estrogen receptor negative breast cancer, but the unexpected finding was that the risk variant, the variant associated with higher risk of the estrogen receptor negative breast cancer, was rather higher frequency in European ancestry populations. And we were expecting the reverse. Then I think that we don't know the answer why, but I think that is telling us is that could be some interaction with environment, some interaction with diet, or we don't know. And I think this type of unexpected result, you have the hypothesis, you expect certain results, but you get a result completely in the opposite direction. And that's it, just open a new avenues of research.
Yeah, and I'm thinking of the opportunities and pitfalls of kind of coming across these changes in science, and how one opportunity is, of course, to just continue along that line of inquiry and open up an entire new world of additional questions to answer. However, one of the pitfalls can be some of the backlash that maybe you'll receive, especially when you think of the peer review process, and then sometimes I also think about the public review process, right? When we report something different from what we previously reported, there's a public response to that. And as they're kind of reviewing and rethinking their theories around it until they can incorporate it into their own perspectives. How do you navigate that exactly? Whether the backlash is coming from the general public or maybe from your peers in your field, when you know that what you found is something that you know that you want to report, but there's a lot of backlash to what you're saying, if you've ever experienced that.
Shanice, I'd like to take this one. So one thing that Dean Bowman talked about in his article is that science is patience and uncertainty, and that means science takes time. And you mentioned that we have, as scientists, a lot of different stakeholders. And one of the stakeholders that scientists need is the federal government, because a lot of the science is funded by institutions like the National Institute of Health or the Environmental Protection Agency or NSF. And we need funding from those agencies to conduct science. And an interesting example I've been thinking about over the last few minutes is how when I joined the University of Michigan 13 years ago now, I wanted to look at the metal lead. as a obesogenic compound. So we knew a lot about lead and neurotoxicity, but there was some emerging data that lead was also changing metabolic syndrome risk and other things like that. And when I put in for these pilot grants and then some bigger grants around 2009, 2010, I was told that lead was solved. We had gotten lead out of gasoline. We had gotten lead out of paint. The blood lead levels in the United States had, dropped, and there was no longer any interest in funding lead. But flash forward to 2015, 16, 17, when we saw the emergence of the Flint water crisis and leaded pipes, which wasn't the first water crisis that had emerged over the past decade and a half. And now all of a sudden, the other stakeholders that scientists want to serve, the community, was asking, where is all the research on lead? And the federal government had to pivot and begin to fund lead again. In fact, at that time, I was receiving a grant to study the chemical bisphenol A and the epigenome. And instead of studying bisphenol A in that grant, which was funded in 2016, I ended up studying lead. And this has shown all sorts of epigenetic differences, not only in the brain, but also in the liver and the heart. So tissues that are really important for metabolic syndrome. And so we also have to think about how something we think that is solved and not fundable anymore, something like a crisis, like the Flint water crisis, could bring it back into a fundable mode again.
Absolutely. And thinking about that, that makes me think of that interaction between politics and science, and also some of those social factors. I'm sure the communities that were unfortunately impacted by that crisis were definitely looking for answers. And here we are. We have a field that hasn't really delved into them in order to answer appropriately. So that can lead to some frustration on the public side. Belinda and Edward, have you all experienced or had any experiences where you had to kind of navigate some either public or professional backlash to your work, and how did you navigate that?
Yeah, can I add to this? Yeah, not maybe public backlash, but also I think rather an interest of from people from scientific aspects. And let me tell you this, the history. This is from my mother. We were talking just a few days ago about these new variants of COVID. And she was interested to know why these new variants, for example, the UK variant, how they originate. Then I have to tell her just a brief class of evolution because she was thinking that maybe the virus somehow know how to mutate and how to is going to have then a more aggressive behavior. Then I have to explain to her, no, this because if there is if the virus is just free on the population, it's going to have more opportunity to mutate. Then then I explained to her, then that's the reason why I need to have, let's say, for example, to control the infection rate, we have to control to get more people vaccinated. And I think maybe other people in the general population are asking those kind of question, how these new variants originate. And then as a scientist, that give us an opportunity not only to educate the public to these, let's say, scientific topics like evolution, but also about the implication for public health, about how this virus, if free in the population, is going to have more opportunity to mutate. And just by chance, some of those mutations are going to be more aggressive and going to be more easily passed from one person to the other. And I think that we as a scientist, we also have that responsibility and to take these opportunities to educate to the general population.
Yeah, and I can actually echo that experience in having conversations with my own community about the vaccine. They're like, why is it changing? Why are there 3 vaccines? Why did they say that we shouldn't wear masks and now we should? And I kind of just kind of approached it as like, that's kind of our responsibility. Yes, we do educate the public, also including our own personal communities. I had like a little session with my family members where they could just ask me whatever question they wanted, and I had to answer the best that I could in order to give them the information they needed to make a decision about vaccination status. So yeah, it's been a very, very interesting journey, to say the least. So Now, because we're all academics, this is the moment we'll be waiting for to talk about something you've been exploring in your research right now that you think could cause us to rethink something we currently believe to be true.
Yeah, I'll just follow up on that hopeful comment that I was making before about the epigenome versus the genome. We all heard earlier and last year in 2020 that the gene editing technology CRISPR won the Nobel Prize. And this is a technology that actually goes in and changes your genetic sequence. And there's all sorts of bioethical issues with that, but it's certainly a very interesting tool to study the mechanisms in the laboratory. But it also turns out that you can harness CRISPR and other genome editing technologies for the epigenome. And so we can now go in and develop technologies to say, what happens if your gene expression at your estrogen receptor alpha gene changes? Does this lead to a negative health outcome? And we may be able to go in and change it so that it's not expressed or it's expressed more. So that gives us some opportunities in the future to help protect against environmental exposures that we might have encountered in utero that over a 70-year lifespan can lead to a negative health outcome. And so it does offer some hope for new clinical interventions and therapies that are targeting the epigenome rather than the genome.
Very cool and interesting work. And I love that you all are talking a lot about genetics and the genome, because I think many of us believe that genetics are just static and they're fixed kind of from birth. So I love that you all are able to get this information out about that there actually can be some changes depending on environmental exposures. Edward, would you like to talk a little bit about your research?
Yes, I want to, now that we're talking about the genome, about genetics, I teach a class of genetic epidemiology during the summer. And one of the goals that I tell my students at the very beginning of the class, first I ask them to write a short essay, what are their goals for the class. But at the very beginning also, I tell them that one of my goals is to just that they are going to learn that genetics is not everything. Because we had this idea that, okay, I have that gene for diabetes, or I have the gene for some other kind of disease. Of course, we have some what we call Mendelian disease, a Mendelian trait, that if you have the particular variants, you are going to have a high risk of that particular disease. However, in many of the population level diseases, what we have is an interaction between genes, environment, and then doesn't imply that if you have a certain alleles that are going to give you a higher risk of disease, still you can manage that risk. You can have a healthy diet, you can do, for example, a healthy behavior, because genes are not destiny. Genes, they are not our fate. And that's, I think, again, maybe something that I want to stress that here, even where we are studying genetics, at least in my case, but this is showed to me that we need to take into account other factors more than genetics.
Yeah, so I already talked about this a little bit before when I was talking about the sort of the race and PCB exposure and telomeres, but I think one of the things I'm most excited about in my work right now is thinking about ways to challenge this notion that what we see in terms of racial and ethnic differences in telomere length or other biomarkers is due to genetics, and really trying to explore what are alternative explanations for that, and thinking about how to to use quantitative epidemiologic studies to think about how race and to demonstrate how race is socially constructed.
Awesome. Well, as you can see, there's a lot of great and interesting research going on here today. But unfortunately, our time has come to an end. That's all for today's discussion here at the University of Michigan School of Public Health on how and why good science changes. I'd like to thank our panelists for the thoughtful and engaging discussion, and thank our audience for joining us. We hope you'll continue these conversations offline.